Hello, my name is Riley O, and today I will be presenting my project toward optimizing ATG dosing, search for ideal ATG exposure. So just a little background. Hematopoietic stem cell transplantation, or HCT, is the transfer of a healthy immune system which can recognize and reject any residual leukemic cells after chemotherapy. Unfortunately, many of the antigens targeted by the donor T cells are not leukemia specific and are widely expressed in different tissues. The recognition and attack of recipient tissue by donor T cells results in graft versus host disease, or GVHD. GVHD remains a major cause of morbidity and mortality post-HCT. Historically, GVHD treatment relied on a combination of immunosuppressive drugs cyclosporin and methotrexate, which reduce GVHD but also increase leukemia relapse because they suppress both anti-recipient, or harmful, and anti-leukemic, or helpful, T cells. Anti-thymocyte globulin, or ATG, containing both anti-T cell antibodies and anti-leukemic antibodies, effectively targets cancerous cells pre-HCT and harmful donor cells post-HCT. It is the first immunosuppressive drug shown to reduce GVHD without increasing relapse. However, despite ATG's beneficial effects, the majority of clinical trials have failed to show improved survival. So why is this the case? ATG pharmacokinetics are highly variable, resulting in variable exposure amongst patients receiving the same weight-based dosing. We measure ATG exposure using the area under the concentration time curve, or the AUC. Underexposed patients here are at risk of GVHD, whereas overexposed patients here are at risk of relapse and infections. Published data suggests that there is an optimal AUC, resulting in low GVHD and low infections, but only a fraction of patients receive this. So our goal is therefore to determine the optimal AUC, that is, the one associated with the highest overall survival, but also the highest leukemia relapse-free survival. We hypothesize that this optimal AUC will balance a low risk of infections with a low risk of GVHD. There is currently no gold standard assay for determining ATG concentrations, so we ran a novel assay to do so at different time points as ATG is infused. We then analyzed this data with relapse and survival information collected from patient charts. For the assay, we simultaneously prepared standards, a series of blank sera. A sera is just a blood component with known ATG concentrations, and patient sera, which contain unknown ATG concentrations. We then incubated these sera with cytochrome cells, which are standardized lymphocytes for the ATG antibodies to bind to. We then washed and stained these samples with a fluorescent anti-rabbit antibody. This antibody binds to ATG. Thus, the more fluorescence in the sample, the more ATG is present. We then ran these samples through a flow cytometer, which can detect the mean fluorescence intensity of a given time point or standard. These standards let us generate a standard curve, which relates fluorescence and ATG concentration. Now, we can determine how much ATG binds to a sample based on how fluorescent it is. We use the ATG concentration at each time point to generate a concentration time curve. From these concentration time curves, we can measure the AUC, or as we recall, the area under the curve, and how quickly ATG is cleared. We then divided our 167 patients into 5 quintile groups according to AUC. So this just means that the 33 patients with the lowest AUCs were in quintile 1, the next 33 lowest were in quintile 2, so on and so forth. Using a multivariate analysis, which takes into account known variables influencing survival, we compared the survival amongst these 5 quintile groups. So what do we find? Survival was highest in quintile group 2, beating the second lowest AUC group. Survival was significantly higher compared to group 5, and what really we can see is markedly higher than any of these other quintiles. And same deal with this survival curves down here. Quintile 2 has a markedly higher survival than these other 4 quintiles. Same deal here. So what's so special about quintile 2 exactly? Compared to all quintile groups, we found that this group had the lowest incidences of relapse and fatal infections, as well as the second lowest incidence of severe GVHD. Exactly what we were looking for in our optimal AUC. So what do? We identified quintile 2 AUC as the optimal AUC. Mechanistically, we think it's because of several factors resulting in low relapse, low infections, so less killing of the helpful T cells, and good killing of the harmful T cells. We will eventually develop an individualized ATG pharmacokinetic model with this data such that every patient will receive the optimal AUC. This personalized approach aims to maximize chances of a favorable survival outcome by tailoring ATG dosages to achieve the optimal AUC for each and every patient. I'd just like to briefly acknowledge my supervisor, Dr. Storik, Hamid Ghazal, Sophia Leuchter, the Const Storik Lab, and the Pure Award for this opportunity. And here are my references. Thank you so much.